Hi guys, welcome back to the second session, and we are continuing from where we left. So we are starting looking at glycerides. Previously, we ended looking at glycerol. So now, under glyceride, we need to know that when we look at lipids, the most abundant lipids that we find in living organisms are actually triacylglycerol. So these compounds are actually of economical form of reserve fuel storage. So we find that when these are metabolized, they actually yield more than twice as much as energy per gram as do the carbohydrates. Remember when we talked about functions, we did explain that actually lipids will provide more energy than carbohydrates. So now when we look at the glyceride or a triglycerol molecule, it actually consists of glycerol, which is joined to 3 fat acids. So glycerol joined to 3 fat acids makes up a triacylglycerol molecule. So tri means 3, and glycerol part there, which is it reviews or gives us information that glycerol is part of the molecule. Glycerol is joined to these fat acids. So remember, we did look at glycerol and it being a 3-carbon alcohol. So it contains 3 hydroxyl groups, which are the OH groups. And then we know that the fat acid is a long unbranched hydrocarbon chain, which terminates with a carboxyl group at one end. It terminates with a carboxyl group at one end. So this is just a review of what we've already talked about, the structure of glycerol and fat acid as well. So for a triacylglycerol to be formed, remember tri means 3, so it means that there are going to be a series of reactions. There are going to be 3 condensation reactions, 3 dehydration reactions that will take place in the formation of a triacylglycerol molecule. So remember, this is a condensation reaction. This is a dehydration reaction. So it means that in this reaction, an equivalent of a water molecule is actually removed as one of the glycerol hydroxyl groups reacts with the carboxyl group of the fat acid. So a molecule of water is removed. And hence, this leads to the formation of a covalent bond that is known as the ester linkage, as already alluded to. So now, since we say that 3 reactions need to take place, during the first reaction, that first reaction is going to give us, or it's going to give a molecule, which is a mono. So it's going to be monoacylglycerol or monoglyceride. During the second reaction, what will be produced is a diacylglycerol molecule. Now, the diacylglycerol is actually an important molecule, important for sending signals within the cell. Sending signals within the cell is an important function that diacylglycerol plays. And in the third reaction, what will be produced is now the triacylglycerol. The triglyceride will be produced in the third reaction. So we said what will be reacting is the hydroxyl group of the carboxyl group of the fat acid reacting with the hydrogen atom of the glycerol molecule, the hydrogen atom of the hydroxyl functional group of the glycerol. These are going to react to form an ester linkage. So remember, we're saying glycerol reacting with the 3 fat acids. So for example, you have palmitic acid, oleic acid, and linoleic acid. You have these 3 acids reacting with the glycerol through the process of esterification, through the process of dehydration or condensation. Water is given out. Water is given out, and this is going to lead to the formation of a triglyceride or triacylglycerol molecule. This lipid, a very important lipid. So remember, we said during the reaction between glycerol and the fat acid, what will be formed is a ester linkage. So you have the ester linkage, and we said that these fat acids that are actually reacting may be different. So for example, palmitic acid, you have oleic acid and linoleic acid. These are different acids reacting with the glycerol to produce this triacylglycerol molecule. Now, when we look at the palmitic acid, it's a saturated fat acid. As you can see, there's no double bond between any two carbons. This is a saturated fat acid. And then when we look at oleic acid is a monounsaturated fat acid. Remember, it's only have a double bond, a single double bond. This is monounsaturated fat acid. And look at the configuration also. Is this a cis or a trans configuration? It is a cis configuration. The hydrogen atoms are on the same side. So oleic acid is a monounsaturated fat acid, and it has a cis configuration. This is a cis configurated monounsaturated fat acid. When you look at the linoleic acid, you're going to realize that it has 2 double bonds. So this is a polyunsaturated fat acid. A polyunsaturated fat acid. So we are seeing here in this molecule, in this triacylglycerol molecule, there are 3 fat acids, each of them different. We have a saturated fat acid, we have a monounsaturated fat acid, and a polyunsaturated fat acid. So the fat acids can be different. So when during the process of digestion, these triacylglycerol molecules are actually hydrolyzed, leading to the production of the fat acids and the glycerol from which they are made from. And as I mentioned earlier, the diacylglycerol is an important molecule involved in sending signals within the cell. So this long hydrocarbon chain, these long hydrocarbon chains that of the fat acids form the hydrophobic chains, or tails rather, which makes the lipids to be insoluble in water. So when you look at the triglycerides, they can be classified as fats if they are solid at room temperature, or as oils if they are liquid as room temperature. So now let's look at the phospholipids. Phospholipids. So these are lipids with a phosphate group. So phosphor is for the phosphate group. Lipids say lipid. So now when you look at a phospholipid, it's the phospholipid is actually among the most important molecules of the cell, because these actually form the core of all biological membranes. So cell membranes will consist of the phospholipids. And these phospholipids also belong to the group of lipids that are called the amphipathic lipids. So remember, I did explain what amphipathic lipids are. So amphipathic lipids have one end of each molecule being hydrophilic and the other end being hydrophobic. Now, when you look at the structure of a phospholipid, it consists of glycerol, which is a 3-carbon alcohol, as we have explained. So this gives or forms the backbone of the phospholipid molecule. The other part is that a phospholipid will consist, or phospholipids consist of fat acids, which we've already explained what fat acids are. These are long chains of unbranched hydrocarbons, long chain of unbranched hydrocarbons, which end in a carboxyl group at one end. So now, in the phospholipid, there are two fat acids that are actually attached to the glycerol backbone in a phospholipid molecule. So two fat acids are actually attached. Then we have the third component of the phospholipid, which is the phosphate group. So the phosphate group is a third component of a phospholipid. So now this is attached to one end of the molecule. This phospholipid will be attached to one end, or rather this phosphate group will be attached to one end of the glycerol. And this group is actually charged. So now this charged phosphate group is usually charged, or is usually having a charged organic molecule which will be joined to it. So the molecule could be a choline, could be ethanol, ethylamine, ethanolamine, or it could be an amino acid, serine. So this is the basic structure of a phospholipid. It is going to contain a glycerol, going to contain a fat acid, and a phosphate group, and remember, due to the presence of this charged phosphate group and also the charged organic molecule, the phospholipids, a phospholipid is an example of a charged molecule. It's an example of a charged molecule. And usually this organic compound contains nitrogen. It usually contains nitrogen. Now this is a structure which is illustrating a phospholipid. Like we said, there are going to be two fat acids. Two fat acids. So we have this fat acid and this fat acid. Remember, these are the non-polar tails. These will be the non-polar tails. And interesting enough, you have two different fat acids here. You have the saturated fat acid and the monounsaturated fat acid in this phospholipid molecule. And then remember I said that the third component is a phosphate group attached to an charged organic molecule, for example, choline. In our case, for example, choline. Yeah. So this part is the head and it is polar or hydrophilic, while this part is the tail and it is hydrophobic. It is a hydrophobic. Interesting enough, we can see when we compare the phospholipid to the triacylglycerol, the phospholipids seem to be a modified triacylglycerol. Seems to be a modified triacylglycerol. Just that one fat acid has been replaced by this phosphate group, which is linked to this charged organic molecule, usually containing nitrogen. Remember, the triacylglycerol do not actually contain phosphorus or nitrogen. So these are very important, as we have mentioned. These are very important structures in the cell membrane. They're important structures in the cell membrane. So now, because they have that amphipathic property that we mentioned, there's a polar head and the non-polar tail. There's a polar head and the non-polar tail. It means that that portion which is composed of the glycerol, this one is composed of the glycerol and the phosphate and the organic, this organic group or the organic base. This portion is actually ionized and readily dissolved in water. So now,
This amphipathic property of phospholipids cause them to form lipid bilayers in water or in water solutions, in aqueous solutions. They're going to cause lipid bilayer. And hence, no wonder they are very suited for their structural presence in cell membranes. So now the phospholipids are also able to form micelles and liposomes in water due to their amphipathic properties. So when the phospholipid is in water, the non-polar tails of the nearby lipid molecules will actually aggregate away from the water, which is going to lead to the formation of these spherical micelles, with the tails facing inwards. So the tails will be facing inwards, as it is illustrated here. They're going to face inwards. So this is actually a perfect example of how detergent molecules work to make grease be soluble in water. So we find that when we look at phospholipids, a more structure actually forms also in which two layers of the molecules are going to line up, with the hydrophobic tails of each layer pointing toward one another. Are going to be pointing towards one another, or also even inward, and this is going to leave the hydrophilic heads, which will be oriented outwards, forming a bilayer, as we have explained earlier. And these bilayers, lipid bilayers, are actually the framework from which the lipid, or from which the cell membranes are actually formed. And then phospholipids can also form the liposomes, liposomes. Now, when we look at the other important lipids that we have are waxes. So waxes are actually esters of fat acids and long chain alcohols. They're very important for waterproofing in both animals and plants. Remember, we gave an example of the cuticles in the leaves of plants, which prevent excess loss of water through the process of transpiration. So they're very important for waterproofing. They're also important for offering an additional protective layer, as I explained, on the cuticle of the epidermis of leaves, fruits, and also in seeds. And they are also found in the skin, fur, feathers of animals or vertebrate animals, and in insects, they are also part of the exoskeleton. They're also part of the exoskeleton. This is the formation of wax. Remember, I said that it is an ester of fat acid and long chain alcohols. So through the process of dehydration and water is given out, we have this ester, which is a wax. It's formed from fat acids and long chain alcohol. So when you look at beeswax, which is a special type of wax found in the honeycomb, to which the bees actually use it for storage of the honey, and also to be able to protect the larval and the pupil during the larval and pupil stages. And it is also used as a glazing agent for fruits, as a coating agent in grease, in cheese and chewing gum, and lip balm and lipstick. So it's a very important lipid in the cosmetic industry, as we have seen lip balm and also lipstick there. It's also used in cheese and in gum. Another example of a wax is the Brazil wax, which is obtained from the palm tree, the leaves of the palm tree. So
It's also known as the palm wax. This is also very important or essential in that it is used in automobile waxes, in shoe polishes, in instrument polishes, in floor and furniture waxes and polishes, in dental floss, and also in foodstuffs, food products such as sweets. So it's a very important molecule. It's a very important lipid. Then we have the steroids. So look at the steroid. It is a molecule which consists of carbon atoms that are actually arranged in 4 attached rings. So it has 4 attached rings. Of those 4 rings, 3 of those rings will contain 6 carbon atoms. 3 of the rings will contain 6 carbon atoms, and the 4th will contain 5. So now, the difference in the length and structure of the side chains that exist or that extend from these rings are going to distinguish one steroid from another. Of important steroids are cholesterol, we have bile salts, we have the productive hormones, and also other hormones that are produced by the adrenal glands. So for example, cortisol. So this is an example of a steroid, and in this case, this is cholesterol. This is cholesterol, which is of biological importance, very important steroid, very common steroid in animals. So it is an important component actually of the animal cell membrane. This cholesterol is a very important component of the animal cell structure and function. However, we know that excess cholesterol in blood leads to the formation of plaques on artery walls, and hence increasing the risk of cardiovascular diseases. And then also cholesterol is important as it acts as a precursor molecule to fat soluble vitamins and also steroid hormones such as the sex hormones. So this is cholesterol. Remember we said that a steroid will contain 3 6-carbon rings. So 1, 2, 3. These are 6 carbon rings, while the 4th is going to contain 5 carbon. So the 4th is a 5-carbon ring. And then we said that what distinguishes these different steroids are distinguished by the functional groups or the side chains that emerge or emanate from these rings. So for example, in cholesterol, we have the OH group here, have the OH there. And then in cortisol, we have the oxygen double bonded to the carbon there. We have this chain here, or this structure in cortisol, while we have this structure in cholesterol. So this is the isoprenoid structure. This is the isoprenoid chain. This is the isoprenoid chain. So this is how you distinguish the different steroids. So you look at the rings. When you look at the rings, they contain 6 carbon, 3 6-carbon ring and 1 5-carbon ring. Then you look at the functional groups. There's groups that are emanating from these rings. This is still showing a steroid, and in this case, this is still cholesterol. Remember, it has the hydroxyl. It has the hydroxyl group there, and then there's the isoprenoid chain. Isoprenoid chain. This hydroxyl group makes it to be hydrophilic or polar.
or this chain makes it to be these steroid rings, and also the isoprenoid chain here makes it to be hydrophobic. So now lipids can also be used as chemical mediators. So animal cells are able to secrete the chemicals that communicate one to another, and also to be able to regulate their own activities. So now some of those chemical mediators are actually produced through the modification of fat acids, which have been removed from the phospholipids or the membrane phospholipids. So examples could be mediators of inflammation. So for example, prostaglandins are some of the chemical mediators. Also have certain hormones also, which are derivatives of fat acids. And the lipids can also act as chemical mediators. Then we also have pigments in plants which are called carotenoids, which are also classified as lipids, as they exhibit the properties of them being insoluble in water, and they also have an oily consistency. So these pigments are actually found in the cells of plants and play an important role in the process of photosynthesis. Animals are actually able to convert carotenoids to vitamin A, which is a very important component for the visual pigment retinol. So no wonder vitamin A is very important for sight. So carotenoids, for example, carrots are very important for sight because they contain carotenoids which are going to be converted to vitamin A. And this is going to lead to a greater and better functionality of the eyes and sight as well. So this is a steroid. In this case, we've looked at the steroid. This is for revisions or as a review. This is the steroid cholesterol. This is cholesterol. You have the OH group there. You have the isoprenoid ring there. This is cholesterol. So having looked at this would mark the end of the session.